It has no brain, it has no neurons, no nervous system, no central processor of any kind. It is, by the strictest biological definition, a single cell. But that single cell can solve mazes. It can recreate the Tokyo subway system in a petri dish. It can learn to ignore irritants, and then teach that learning to another slime mold by physically merging with it. Its name is Physarum polycephalum, the many-headed slime. And what it can do has forced biologists to ask a question they didn't expect to be asking in 2026. If memory can live without neurons, what exactly is memory? In the year 2000, a Japanese biologist named Toshiyuki Nakagaki did something most scientists wouldn't have thought to try. He took a single slime mold. He chopped it into pieces. He scattered those pieces throughout a plastic maze. Then he placed 2 blocks of oat flake agar, one at the start of the maze, one at the end. The slime mold pieces did what slime molds do. They reached out for the food. They grew tubular extensions in every direction, exploring the corridors of the maze. After about 4 hours, the slime mold had filled nearly every passage, connecting one food source to the other through every possible route like ink filling a glass pipe network. And then, and this is the part that broke people's brains, the slime mold began to prune itself. It withdrew from the dead-end corridors. It collapsed its extensions in the longer paths. After 8 hours, the slime mold existed only along the shortest possible route between the 2 food sources. In 14 out of 19 experimental trials, the slime mold chose the shortest path, or the second shortest, usually within 2% of optimal. A single cell, no brain, no eyes, no way to see the whole maze. And yet, it solved the maze. How? The mechanism involves something called protoplasmic streaming. The slime mold's body is a single continuous cytoplasm, like a giant amoeba with many nuclei but no internal walls. That cytoplasm oscillates, it pulses. Different parts contract and relax in waves, pushing fluid through the tubular network. When a part of the slime mold finds food, the chemical signal triggers stronger oscillations in that direction. Tubes carrying more fluid get reinforced. They widen and persist. Tubes carrying less fluid weaken. They thin and disappear. Over hours, the network reorganizes itself toward the shortest food-connecting route. It's not thinking, it's not planning, it's just flowing intelligently. 10 years after the maze experiment, Nakagaki's collaborator Atsushi Tero asked a bigger question. What if the maze wasn't a maze, but a city? In 2010, Tero and his team laid out oat flakes on a wet surface. The arrangement wasn't random. Each oat flake corresponded to a real urban center in the greater Tokyo area. Yokohama, Chiba, Saitama, Hachioji. 36 cities total. Then they placed a single slime mold at the center, the position of central Tokyo. The slime mold did what slime molds do. It explored. It connected the food sources. It pruned its network. After 26 hours, the slime mold had built a network of tubular connections linking all 36 oat flakes. When the researchers compared the slime mold's network to the actual Tokyo rail system, built by engineers and city planners over more than 100 years, they found something remarkable. The 2 networks were nearly identical. Same major hub connections, same redundancy patterns, same efficiency versus cost trade-offs. The slime mold had recreated a transportation system that engineers had spent generations refining in a little over a day, without a brain, without engineers, without a single meeting. The researchers published their results in the journal Science. The paper was titled, almost casually, Rules for Biologically Inspired Adaptive Network Design. Translation: maybe we should be paying attention. But the maze and the subway, those are about navigation, about finding the best path through space. Impressive but explainable. Chemistry, flow, feedback. The real surprise came in 2016. A French biologist named Audrey Dussutour, working at the University of Toulouse, asked a different kind of question. Can a slime mold learn? The setup was simple. Slime molds normally crawl freely across food sources, but Dussutour put bitter substances in the path, caffeine, quinine, substances that slime molds normally avoid. A slime mold encountering caffeine would slow down, hesitate, sometimes refuse to cross. Dussutour exposed the slime molds to caffeine over and over, days in a row, and something strange happened. The slime molds stopped hesitating. By the 6th day, they crossed the caffeine bridges almost as fast as plain water. They had habituated to the irritant. They had, in some sense, learned that the caffeine wasn't going to hurt them. This is habituation, the simplest form of learning. It's how your nervous system stops registering the feel of your clothes against your skin. It's how a sea slug stops flinching when poked in the same spot repeatedly. But sea slugs have neurons. They have a brain, small but real. Slime molds have neither. And then Dussutour took it further. She let the habituated slime molds dry into a dormant state called sclerotia, like a slime mold hibernating. She kept them dry for 1 month, then she revived them with water. The revived slime molds, having gone through complete dormancy, having essentially shut down all metabolic activity for 30 days, remembered. They crossed caffeine bridges quickly. They retained the habit. And then, the most unsettling experiment of all, Dussutour took a slime mold that had been trained to ignore caffeine. She took a naive slime mold that had never seen caffeine. She placed them next to each other. Slime molds, given the chance, will fuse, merge their bodies into a single larger cell. After the fusion, the formerly naive slime mold could now cross caffeine bridges without hesitation. The learning had transferred, through cell-to-cell contact, from one organism to another, without any neurons, without any signals you'd recognize as communication. Just body-to-body merger, and the memory came with. Here is what biologists are now forced to consider. For most of the last 100 years, we have assumed that memory lives in neurons, specifically in the strengthening and weakening of synaptic connections between neurons. The trace of an experience is stored somehow in the wiring pattern of the brain. This is the engram, the physical substrate of memory, the holy grail of neuroscience. But slime molds don't have neurons, they don't have synapses, they don't have wiring, and yet they remember, and yet they teach each other by merging. This suggests that memory, at its most fundamental level, may not be about neurons at all. It may be about something more basic: chemical signaling cascades, protein modifications, cytoplasmic states, networks of feedback that any sufficiently complex cell can maintain. If that's true, and the slime mold evidence keeps pushing in that direction, then memory is much older than brains. Memory may go all the way down, to single cells, to bacterial colonies, maybe further. We are not the only things that remember. We may not even be the most efficient at it. A slime mold can do in 26 hours what took city planners a century. It can learn, it can sleep, it can wake up, it can give its memory to another being by touching it, and it has no idea who it is. That's the part I keep coming back to. The slime mold doesn't know it solved the Tokyo subway. It doesn't know it learned to tolerate caffeine. It doesn't know that another cell now carries its memory. It just does. Maybe consciousness, the knowing that we know, is a much later invention than memory. Maybe consciousness is what brains added on top of something the rest of life had been doing for 1 billion years without us. The slime mold doesn't need to be conscious to remember. The question is, do we need to be conscious to be alive? I'm Sebastian. This was Apocrypha and Octopuses. Thank you for sitting with the slime mold for 9 minutes. The papers behind this video are in the description: Nakagaki 2000, Tero 2010, Boisseau and Dussutour 2016. Read them slowly.